Okay, great. So now I'd like to introduce our speaker for today. Mike Davis is an aquatic ecologist with the Minnesota DNR. As a child, Mike was enchanted with water, and he's worked on and under rivers ever since. He's been a commercial fisherman, a fur trapper, a clam harvester, and a dairy farmer. And he found his clam harvesting years so interesting that he decided to return to college, and in 1986 he graduated from Winona State with a BS in biology. He began working for the DNR in 1987, and there Mike has worked on mussels for decades. First on a mussel survey of the Cannon River system, then by starting a statewide mussel survey program, then on propagating and releasing Higgins eye mussels into the Mississippi River, and then more recently by establishing the Minnesota DNR Center for Aquatic Mollusk Programs, where they've expanded the propagation to 10 additional species of native mussels for reintroduction. So, in a nutshell, Mike knows his mussels. So we're really happy to have him here today, and you can take it away, Mike.
Okay, thank you. So the beautiful picture of mussels is from the St. Croix River, and a lot of our inspiration for trying to reestablish mussel species and populations of mussels in other rivers comes from the amazing biodiversity of the St. Croix. That was our initial inspiration, I guess you could say. Today I want to go over first the life history of these animals. What makes them interesting is how they live and what they do when they're living. I'm going to also cover impacts humans have had on them, particularly since European settlement, which is what we know the most about, of course, and why mussels are important to people and the ecological health of rivers, and how that relates to clean water, which is a huge goal of the state, and to aquatic habitat that they complement for other animals. And then I'll give a summary of our program and what we're trying to do. So getting into this, I don't know if you can see my mouse that show up.
Yeah, we can.
Okay, so I'm pointing at a fat mussel. This is sort of the basic generic mussel of Minnesota. It's found in every watershed of the state. It has a little foot that it pushes out into the sand, and when it pushes it out, inflates it, and when it pulls it back, it moves its shell ahead, leaving a little trench. You may see these along the shore of a lake or in a river. But they can't move very fast or very far that way. Their main method of dispersal has to do with their life history that I'm going to go over next. But this picture is a mussel in a riverbed. This is where it pulls water in from the river, which contains oxygen and the food that it eats. And then this other little opening is where what it doesn't want goes back out. The water flows back out again. And these are not zebra mussels, so don't mistake them for that. This picture is a native mussel covered in zebra mussels in Lake Pepin. They can be a problem. They can suffocate our natives, so we don't particularly like having them show up. Mussels come with a lot of different shapes and colors, and they've acquired some really colorful names to go along with that. You can kind of read them on the screen there, so I don't need to go over them, but we have about 52 species in Minnesota that we know of, or used to have anyway. And they all have their own unique names. A lot of those names are given to them by pearl hunters and people harvesting them for shells. The life history, though, is what's most interesting. We have male mussels and female mussels, and the males fertilize eggs within the female, and they produce a larval form that has to spend the time as a parasite on a fish. So typically they clamp down to the gill tissue or even fins of the fish, and the fish will embed those little larvae into their tissue and feed them. Here's a microscope picture of them encapsulated in the gill tissue. They'll spend 2 or 3 weeks on a fish. Meanwhile, the fish swims around, and that's really how they get moved up and down rivers and around lake shores, is by the fish hosts that carry the larval form. Of course, there's a challenge in all this. If you're in a big flowing river like the Mississippi, how is that male going to get the sperm to the eggs in the female? The female has an ovary like many animals, and those eggs are stored in the little tubes in the gill chambers of the female mussel. Little diagram there to kind of show that. So the trick is, with thousands and thousands of eggs in each gill chamber, how do you get them fertilized? And here's a spermatozygmata from a male mussel. You see it spinning around with all the little tails flying out of it. So these are what we call sperm balls for short, and they contain thousands of sperm cells. And when a female mussel has one of these sperm balls of the right species drawn in through her incurrent siphon, it gets a chemical cue of some sort and explodes literally into thousands of sperm cells that then go ahead and fertilize eggs. So it doesn't require a whole lot of these to get all your eggs fertilized to produce larvae. And the genetic advantage is that there can be multiple male parents in one brood of larval mussels from the female. This is a dissection showing those tubes of a mussel that are full of the little larvae, which under a microscope kind of look like this. When they leave the female mussel, they're open. When they encounter the fish's tissue, they snap shut. And we can stimulate that to happen with a few grains of salt under the microscope to check them for viability in the work we're doing. So the next problem for the mussel is how to get those larvae onto the fish. And there's a lot of really interesting ways that this happens. And they usually involve trying to entice a fish to the mussel by pretending to be a food item. In this case, this is a mussel, fat mucket in the Chippewa River. And I'm going to play the video. This is under the eye spot. This is all muscle tissue simulating a minnow. And inside right here are the gill chambers full of larvae. So when the fish comes in to strike that, instead of getting the meal it's looking for, it gets a mouthful of those larvae that quickly start attaching to its gills. And here's another species, not from Minnesota, that also pretends to be a food item. That look like a crayfish to you? I think it did to me. And this is yet another one. You've heard of fishing with Rapala lures? This mussel has a Rapala lure that releases out of its shell on a transparent or nearly transparent monofilament line, so to speak. And that little lure will swim around and attract fish that bite it, thinking that it's food. And then these chambers that it somehow is released from its body are still full of the larvae that clamp onto the fish. So some species don't use big predators like walleye and bass, like the ones I just gave examples to. Some use minnows. This is a monkey-faced mussel in the St. Croix River. You watch this structure, it's the excurrent siphon of that mussel. And when you see it suddenly retract, you'll see a swarm of stuff go out. It's shooting and literally shotgunning its larvae, usually into a school of minnows that it somehow detects as they approach. Pretty cool stuff. So what we don't see above the river on the beautiful St. Croix, like this picture is, if you go beneath the surface, there's all kinds of crazy stuff going on down there. This is a purple warty-back mussel. As I settle down a little. And this is a conglomerate of its larvae that it's extruded from its shell. Now, these mussels use catfish as hosts, and we think this probably tastes good, stimulates food items for a catfish browsing along the bottom. And as it bites in on that, of course, it gets the larvae on its gills and swims off with the babies again. So those are the ones that try to bring the fish to them. Others send out little packets for the fish to find. They call them Trojan horses. I think that's a great name. This is an example of a mussel actually in Missouri that has a great simulation of a black fly larvae. Black fly larvae look like this. They have a sticky end that's usually stuck on a rock. And this mussel produces packets in its gill chambers that resemble these larvae, only on the inside are all these little kitty larvae. And when the fish bites them, it breaks open that body and out come the babies. So the next slide is that actual video of that going on. Darters are the hosts to that mussel. This darter is about to feed on what it thinks is a black fly larvae, but then it spits it out right away. Oh, I was going to play it again for you. You can see the larvae that's swarming around its mouth right after it spits it out. So once they've been fooled, they've kind of been had by the mussel, because then they swim off. So the last technique is the one that actually captures its fish, ambushes the fish. This is the snuffbox mussel. It's a fairly endangered species. We still have a nice population in the upper St. Croix River, just below the dam at Taylor's Falls. And Their method is exploits the feeding habits of the log perch, type of darter that is the only viable host for the species. And the log perch feed by flipping over pebbles. So the female is pretending to be a pebble, only her shell is agape there, and it's actually a trap. So along comes the log perch to flip that pebble, and whammo, suddenly slams shut, got little serrated teethy edges on its shell, and that fish can't get away. And the next thing it does is shown in this video, where it starts to pump its shell open and shut, and you'll see larvae flowing out. And it's pumping mussel larvae through the mouth of the fish and across the gills where they're attaching. And after a half hour or so, it will release that fish to swim off with its babies and do its part. So there's yet another method. We discovered freshwater drum, or sheep's head, are host to about 6 different mussel species in Minnesota. This is a deer toe mussel that we discovered by diving in the dark on the St. Croix River and never been reported to science before. Bernard Seidman and I plunged under the water one early spring. But this video shows what happens when you touch the mantle tissue here of the mussel, simulating a fish coming along. And what they do is it open up, and there's all the larval gill chambers. So if you're at freshwater drum, you know mussels taste good, and you're equipped to eat mollusks because you have teeth in the throat of your throat that can crush shells. But if it's a big enough one, they'll still try to eat them. But we think when they bite onto them, they pull out larvae and get inoculated and swim off. Then this is a pink heel splitter, another mussel that uses the drum. Similar attraction, we think, in these mantle tissue, and there's the larvae ready to spew forth into the fish's mouth. And of course, this is a federally endangered Higgins eye, and there's those gill tissues pushed way up in the air above the fish lure. Pretty cool techniques. So once they've been on the fish and transformed or gone through a metamorphosis, they come off as these little tiny guys, juvenile mussels. They have a little bit of a foot that they can crawl around with, very sticky foot in most species. And then they grow a tether line, which you can barely see probably, but it anchors the little mussel by tangling up on rocks or sticks, and like a safety tether so it won't be washed away. Then they grow quickly when they're young until they reach sexual maturity. And depending on the mussel, that can take anywhere from 1 to 7 years to reach maturity and start reproducing. So the thing about mussels is they're the most endangered group of animals in North America. Probably around 300 species in North America. But mussels and snails are at the top of the list of how many species are endangered. And really the first 4 categories here in this chart are all water-dependent animals. And so water magnifies the impacts of people, of course, because it comes off the landscapes and things get concentrated. But in the conservation world, of course, we spend most of our time working on birds, mammals, and butterflies, and visually obvious species. And it turns out fewer species of those categories are endangered. So we're trying to change that around a little bit, not to stop work on birds, mammals, and butterflies, but to increase our awareness of the water-dependent animals that are at most risk. So human history goes way back. The native folks in North America used the shells of mussels for ornaments and tools, and of course as a food source. So when people discovered you could find a pearl in one occasionally and get rich, people started harvesting them. The Europeans started harvesting them from rivers. And shortly after that, a guy from Germany figured out you could make buttons out of their shells. And so we had camps along the river. When people would go out in their boats, they have these chains with little hooks on the ends. They drag them across the bottom, and the mussels that are feeding will find that a disturbance of that hook dragging over them, clamp shut, and then they can pull them up. And by doing that day after day, they were harvesting thousands and thousands of animals. In fact, they're so abundant in places on the Mississippi, they would go out in the winter, cut a hole in the ice, and with a long-handled rake, scoop up the shells or the live mussels and pull them up on the ice. And here you see them in the Mississippi down in Iowa. Piles of shells all over the ice, which gives you some indication of how abundant they once were. And think of all those thousands and thousands of animals filtering water and removing particles and nutrients and contaminants from the river. Pretty astounding. We think in some places probably 300 to 400 animals per square meter. Barge loads of shells were shipped from all sorts of rivers, not just the Mississippi, but anything that you could get into that had mussels, which really ranged from decent-sized creeks on down to on up to the size of the Mississippi and St. Croix. So the result of that were feeding the button factories. It was a typical tragedy of commons, like the sardines in Monterey Bay. The resource couldn't handle the amount of harvest going on. Factories then were employing thousands of people across the Midwest. That got to be an issue for the biologists because they were running out of mussels, making these buttons out of them. So that and pollution, which sort of coincided at the end of the button harvest, was diminishing the river's ability to restore the mussels anyway. These are sewage mats in the Mississippi and St. Paul, 1933, not that long ago. I guess it's long enough, but fortunately we don't see those anymore. We've made a lot of progress. And other impacts, from clearing the land, forests and prairies, and erosion of the soil from cropland, and then the increased water flow into rivers, destabilizing the banks, all work to create unstable habitat or contaminated habitat for mussels and other critters. And then if that weren't enough, we built dams on most of our rivers. So remember, the mussels are dependent on fish to swim around with the larvae to keep them distributed or to recolonize areas from which maybe they suffered. So if there was a pollution event up here that killed all the mussels, and the fish there's still mussels downstream, the fish can no longer get there to bring the mussels back and recolonize. So that's become a big issue for mussels as well. So we think they're important for indicators because they're long-lived animals. Some species live to be 100 years old or more. And unlike a fish or a bug, they can't swim away or fly away to escape pollutants that may enter the river. They have to either take it or die. And the ones that die leave their shells behind. That provides a reference condition we can use for trying to figure out what species we might want to try and reintroduce into the river. And as mussels filter water and particles, they actually can sequester contaminants in their shell each year that they're growing more shell. Mussel aggregations can become dense, as I said before, forming habitat. They process the nutrients they take in and create a microcosm of fertility in rivers that other insects, aquatic insects and so forth, thrive on, and remove contaminants in the process. And the big reason most people can relate to is that fish love mussel beds and hang out in mussel beds because there's a lot of food there. There's a pile of shells near the Mississippi near Red Wing along the river, and there's a huge pile from a pearl hunters camp. And of course, that was my first introduction to how many species were missing in the Mississippi, because we could identify all the shells in that pile. There were at least a dozen species that were common in the pile that were no longer found in the river. So the annual growth of a mussel can be seen in between these lines, like tree ring lines. And within each year, there's a little micro history of what came down the river contained. And that's actually been used to nab polluters that have created problems for other animals. Usually it's fish kills that alert people, but there was a case out in the east where a mining company was convicted in court based on the records of the contaminants they released in mussel shells downstream of the mine and upstream of the mine. So it's a pretty useful thing to be able to use. So this idea of mussel being habitat, I think, is illustrated nicely by this photo of a mussel in Mille Lacs Lake, where caddisflies are grazing the algae that grow on the shell of the mussel. Those algae are receiving the nitrogen phosphorus mix from the mussel secretions, or excretions, I guess I should say, that create a chemical area that seems to favor more species of algae and diatoms. And then over on this side, we have a caddisfly grazing with a snail grazing the algae that's growing on the back of the caddisfly, and a hydra perched on the snail shell capturing the debris flying around in that little microhabitat area. So begin to see life building upon the building structures of the mussels, and I'm reminded of coral reefs, and I like to think mussel beds are the coral reefs of fresh water, or they can be when they become abundant enough. Part of that is that they're, as we said, they clear the water. And this is illustrated in a little test that was done out in Virginia. So kind of dramatic when you can suddenly see the trees through the water again. And if that's too hard to imagine the bottom of the river, you can just think of them as a cartoon, little bunch of critters down there cleaning up our rivers. Busy guys. So this picture shows Lake Pepin and the foot of Lake Pepin where the river recollects into actually it becomes a river again. And these are all fishermen fishing over one of the highest density and species-rich mussel beds that we've ever found in the Mississippi River. One day there were 26 boats fishing, drifting along over the mussel bed. Pretty dramatic. So there again, this is that nutrient capture and cycling, producing food. Mussel or small fish are attracted in to feed on the aquatic insects, and then big fish feed on the little fish. The mussels are there releasing larvae to inoculate the fish that stick around and drop the transformed juveniles back into the mussel bed. So it's kind of a self-reinforcing positive feedback loop in the aquatic version of a coral reef. So as the mussels were disappearing, the U.S. Bureau of Fisheries got into the business of trying to propagate and release mussels to provide material for those button factories. And one of those early efforts was on Lake Pepin. These are tanks of fish that are awaiting inoculation by mussel larvae, and that was done by extracting the larvae out of a female mussel. In this case, they just killed them and extracted all the larvae, put the larvae in the tubs with the fish, and then afterwards put the fish into pens out in Lake Pepin. So this is a chicken wire pen or some kind of wire pen, and at the bottom is a big wooden platform with sand in it. And they put fish in here that had the mussels on their gills, left them in for a couple of months, and then released them. And later in the fall, well, they actually put dozens of these big pens out in Lake Pepin at one time. And then in the fall, they used a hand crank winch with a line out and pulled them back up the shore to see what they got. And they were quite successful, actually. So here's those little bissel threads on each one can tangle up, and especially in a confined place. So this is a big string of fat bucket mussels that were one of the preferred button mussels back in the day, all from Lake Pepin. So that was kind of inspiring to learn the history of that. You know, the history of water quality in the Mississippi was pretty dismal. We had the first dead zones, floating sewage mats. And a survey in the late 20s showed almost no oxygen, even as far downriver as Red Wing. So Lake Pepin was in great jeopardy, and they actually abandoned those efforts to try and restore the mussels of Lake Pepin because of the severe pollution that was going on. But thanks to the Clean Water Act and efforts by our Pollution Control Agency and Met Council and their sewage wastewater treatment plant improvements came, and by the mid-'80s, we were meeting our water quality standard again for oxygen. And not only that, but the ammonia levels in the river sediments, the river dropped below an area which we now know is sort of a maximum that they can handle, that the mussels can handle and still live. So conditions returned, but about the time that was happening, the zebra mussels showed up, jeopardizing mussels again. And because we have a federally listed species in the Mississippi, the Higgins Eye, Fish and Wildlife Service issued a jeopardy decision saying that the Corps of Engineers was responsible for bringing zebra mussels upriver from down, because zebra mussels can't attach to fish and swim. They have to be brought by people. And the Corps of Engineers agreed to fund a program, a 20-year program, to try and restore Higgins Eye mussels in areas that didn't have zebra. And so we started on that in 2001, collecting female Higgins Eyes here, extracting larvae with a syringe so the female could survive, and then inoculating the host fish. In this case, it's a bunch of walleye. Push put fish in cages that we sunk to the bottom of the river so our jet skiers wouldn't run into them. And then, a year later, here we have a similar string of baby Higgins eyes that they had in 1919. So we've kind of tried to duplicate their effort, and we're successful. So it's been a great thing. And 10 years later, our monitoring program shows that where we were releasing them at Hidden Falls Park in St. Paul, the population we established was reproducing on its own, which is sort of the ultimate goal of any restoration process. Hopefully that will continue into the future. But that led us to establish a more formalized program at the DNR. It started out with statewide surveys in 1999 and continued on through the years, and now we're doing more propagation. We have 5 permanent employees shown here, also a temporary technician that works for us, so 6 people working today here. And also we hire interns each year to do our dirty work, brave souls that we hope will survive the summer with us. Yeah, but in the process of this last 20 years, we've sampled almost 4,000 sites around Minnesota. Every major watershed has mussel data for us. We know a lot about the distribution of species, where they're abundant, where they're not abundant, and even where they used to be abundant from those shell records. We've set up 16 long-term monitoring sites where we do intensive data collection, geospatial surveys, so we can do population estimates of the reach of river and follow them over time, looking at each species' reproduction, growth, and survival for the long haul. And some of the results have showed that the agricultural watersheds, of course, are the ones missing the most species. The Des Moines River sticks out as a really tough one. The percent of species extirpated reaches, 70-80% in some of those sites. The Minnesota River is in tough shape. Mussel abundance is shown by a catch per effort, mussels captured per minute, for example, plotted here. And again, the agricultural areas have the least abundant, where our St. Croix River is really great. The North Shore shows a low abundance, but it always has. It's cold water streams, and mussels don't really do well in cold water streams. But in 2014, we established this Center for Aquatic Mollusk Programs, which I'm calling. I invented an acronym, and then from the acronym came up with a name. So I sort of cheated, but anyway, it's nice to be able to say we're headed back to camp at the end of the day. So our program objectives really are to try and, at this point, using the information we've collected over the last 20 years, restore historic mussel species aggregations and their ecosystem functions to our streams. And in the process, hopefully we can delist some of the state endangered and threatened species so that their abundance returns to a level we think that they're no longer in jeopardy. And part of this, of course, involves what I'm doing today, trying to inform the public about the unique role mussels play in aquatic ecosystems that really benefit people, teaching the next generation to continue caring for the rivers that we really have improved dramatically over the last 100 years. So part of what we do here is research to determine which fish serve as the best host for the mussel species we're going to work on. And in our lab, we have this system of tanks called aquatic habitat type of things. And each tank, then we can put a different species of fish or replicates of a species, and then inoculate them with whatever mussel we're working with. And we collect what comes off of those tanks onto a little screen, take it to a microscope, do counts, painstakingly document what comes off of that fish. And in some cases, they slough off all the little larvae within a couple of days. And in other cases, they'll carry most of them through full transformation. We know then that's a great host. Here's the freshwater drum and the 6 species it's serving as hosts for. So we would be really happy if the freshwater drum was no longer considered a rough fish and not protected at all by state law. Maybe we can get there someday. The other part of this whole thing allowed us to identify the host to a federally endangered species that we and other labs around the country had worked on for about 20 years. It's kind of an interesting story because we have a really great population of spectacle case mussels in the St. Croix River downstream of the dam at St. Croix Falls, and we have an aging and dying out population of them upstream of the dam. So Bernard and I put our heads together and figured, well, whatever the host is for this must not live above the dam, but used to. So what are the museum records of fish above it? We came up with a few possibilities, one being the American eel, which we tested and didn't find anything, and the other being mooneye and goldeye, which are in their own fish family, separate from all others, the Hyodontidae. And those, they have tongue fish is the sort of Latin transformation. They have tooth tongue, they have teeth on their tongues. But at any rate, they're really hard to keep alive, so they're the kind of fish where you look at them, they turn over and die. So we had to devise a way to keep them alive long enough to test them. And the first year we tried circular tanks that were about 300 gallons in size. And before they could transform anything, they all died. But we saw some promising retention of the mussel larvae on their gills. So with a little help from the Minnesota Zoo's aquarium people, we put in a 1,000-gallon round tank with a bubble curtain around it to keep them from bumping into the sides. And we were able to keep them long enough to get these juveniles that you see over here to live there, or to live. So we know they're the host, and now we're starting to work on conservation, restoration for that species. So, trying to reestablish these ecosystem goods and services is probably not an easy task, and not all rivers are probably in good enough shape to do that, but we have identified some to work on, and that's what we're doing now. So in our lab today, we keep collections of host fish in troughs, feed them till we're ready for them, and then they get placed into a tub like this, along with a beaker full of mussel larvae. We time them and inspect them, and when their gills look like they have enough on them, they're put into a recovery system where each tank holds fish. The water circulates through a sump system, and the water coming out from the bottom of the tank runs through a screen where we can collect juvenile mussels as they transform. Once we've collected them, they go into various culture techniques, tubs with sediment and air where we feed them filtered river water and a mixture of algae that we can buy from a supply house. The idea being to raise them up till they're big enough to put out for more growth in a lake somewhere. So one of our reintroduction opportunities that we identified was in the Cedar River downstream of Austin, Minnesota. Austin being the home of Hormel meatpacking. And, like everyone 100 years ago, everything went to the river. And today, Hormel says that the only thing they throw away from a pig is the hoink. And they really have cleaned up the Cedar River. We have great populations of golden crayfish in there. It's the main river system in Minnesota with that crayfish species. And there's substrate that is pretty stable downstream of Austin. There's a dam at Austin that probably plays a role in that. But there's also a population of 6 or 7 of these species down in Iowa that we can get females from. But there's 3 dams in between the Minnesota stretch of the Cedar and the Iowa stretch, and they can't get up here by fish. So we're intervening, and we're trying to bring them back to the Cedar River. And so part of our effort here was to start producing black sand shells. That's a fairly easy mussel to work with. These are juveniles that were produced the first year we tried in July of 2016, moving around under a microscope slide. And then in April of 2017, they had grown to a pretty good size. And then in May of 2017, we put them out. This is how big they were. We put them out into totes in the East Side Lake, which you'll see next year. We bought totes at Fleet Farm, drilled them full of holes, and then we put a lid on, some sand in the bottom, attach them to these little platforms, and then take them out into the lake and put the mussels in there to grow. And then we come back later, a year later, pull them out, and they're big enough now. These are black sand shells, and we're glue little yellow tags on them so we can monitor their growth, and then place them into grids in the river. And we had enough this year that we put a few hundred in a grid, and the rest we scattered along in the river. These are actually muckets being released into the Mississippi River at Hidden Falls, and also snuff box mussels being released. So we're working on multiple species to do this, and we'll hope to continue doing more. This is a snuff box babies moving around. they actually are fairly viable. We think we got a good chance of getting them reestablished in more places than the St. Croix where they used to live. And so in the future, we're going to expand it, hopefully, with funding. If we can get the funding, we'll expand our propagation restoration to include more species and more streams, and make mussel restoration a part of stream restoration projects where channelized portions are being re-meandered and so forth. So we hope that in the future, mussels will be included in watershed assessments and monitoring as part of the 10-year cycle of deciding how streams are doing, and as we work on restoring watershed function. So hopefully in the future, by 2050 maybe, we'll have the biodiversity, at least in some of our rivers, restored, and the ecological functions, cleaning water for us and removing, as I mentioned, E. coli is a favorite food of freshwater mussels. Recent research shows that they eliminate enormous amount of E. coli bacteria from the they filter. So they have a lot of potential to give us cleaner water. Well, I guess I've gone kind of fast. Our funding is coming from the Environment and Natural Resources Trust Fund through LCCMAR grants. We get federal money from the State Wildlife Grant Program, both competitive grants where that Cedar River project's a joint project with the Iowa DNR. The Corps of Engineers funds monitoring of the Mississippi River mussel beds that help us monitor what we were introduced, as well as the Higgins eye. And then we collaborate with support from other agencies. We've done work for DOT, USGS. We completed a big project this last summer collecting data on mussels from pools 8 and 13 in the Mississippi River for USGS to use in their research. We work with the Minnesota Zoo. They're starting to be able to raise mussels in one of the zoo lakes. So we'll provide them with young mussels to put out in their lake to grow so that they get big enough to release without fear of being eaten immediately by predators. University of Minnesota collaborates on our host research stuff, and the National Park Service has assisted us with surveys and so forth. So a lot of work with other agencies, including Wisconsin, Iowa, and Illinois DNRs. That concludes the talk, so I guess I could field questions at this point if anybody has any.
Yes, so if people want, you can feel free to unmute yourself and ask over the phone, or you can chat. You can type your question into the chat, and I can read it out loud. So whatever you guys prefer. Ask away.
I'll put the pretty picture back up. Don't be bashful.
While people are thinking, I'll ask one of the questions that a volunteer sent me over email. He wondered if he should be trying to kind of rescue the native mussels when he sees zebra mussels attached to them. Should he be scraping them off? Is that going to be helpful to the native mussels? What should people be doing when they see that?
Well, it certainly helps that individual mussel, but it's logistically difficult to clean thousands of mussels frequently enough. We did an experiment actually in the 90s when they first showed up in Lake Pepin, and we had a bunch of tagged mussels that got clogged up with zebra mussels. And we cleaned them all off early in the summer and threw them back into our little grid where we could recover them, and went back in the fall and early October. And they had recolonized with zebra mussels to a pretty large extent. So, but that doesn't mean it didn't benefit those mussels, because that gave them a reprieve where they could reproduce potentially, or at least gain some weight back from being denied their food by all the zebra mussels on them. So I wouldn't see it as a waste of time, but don't expect great restoration to happen from that either. Sort of personally gratifying, though, I have to say.
Definitely. Another question that somebody had was about, they had said that some zebra mussels have unexplainably died off in the St. Croix, and then apparently there's a boat barrier that's now being unmanned for about 7 years. They were wondering if you know why the Park Service hasn't opened the St. Croix up to boating past Arcola Bridge.
Well, I think part of it, number one, probably is because boats still enter the St. Croix from down on the Mississippi, where zebra mussels are still fairly abundant. And they haven't totally disappeared from the St. Croix. We find a few there still, so there's still a possibility of carrying them upstream, and I suppose that's why they don't they don't open it up, but, maybe it's not enough of a crisis to put the funds to manning that station they used to man.
Mm-hmm.
We have another reason. Our biggest fear is that they'll get it established in the reservoir above the dam someday and then rain larvae down on the river below. So I don't really know the answer. You'd have to ask the Park Service. I'm just speculating.
Feel free, if anybody has any questions, you can type them in the chat, or you can unmute yourself. Maybe I missed this, but how many mussel species did we have, like total, before we kind of saw the big die-off today? Did you say how many?
Well, I think historically there were probably 52 in Minnesota.
Okay.
And we've lost a few of those. They maybe never were abundant, but there are museum records of a few. And there's some that we think probably are extirpated now, but although they haven't been listed as such, they're still listed as endangered. Yeah, so today, the Mississippi River below St. Anthony Falls probably had 42 or 43, so did the Minnesota River. We think the St. Croix was up in that neighborhood of 40. But they're not all occurring at one spot at one time typically, but there are, the area right below St. Croix Falls Dam has around 30 species. It's really cool to see so many different ones in there. And some of the rarest ones are still hanging on there. We have a couple that have disappeared and we think are extirpated that are dependent on a migratory fish, the skipjack herring. When the first dam on the Mississippi was built at Keokuk, Iowa, it blocked the migration of those fish, and they can't survive winters in the north, so they would swim to Minnesota to spawn in the spring, and then swim in the fall, swim back downriver where it didn't get so cold, survive, and now they can't get upstream past that dam, and the populations that apparently went on downstream and never came back. And so there are two species, the elephant ear and the ebony shell, that depend on them as a host fish. And we last saw a live ebony shell about 10 years ago at Prescott, Wisconsin, and the last couple times we've tried to find them again, all we find are the empty shells. So we think that it's been over 100 years since the dam was built, and they finally just died out. But there's no official acknowledgement of that so far. I'm sure there are others that are affected by the lack of movement through the dams. One species made a miraculous return. The warty-back mussel was once fairly common throughout the Mississippi and Minnesota and the Minnesota River. And they sort of disappeared until sometime after the 93 flood, I speculate, because their host is channel catfish. And in 93, during the brooding period for that mussel, which is summertime, well, we had a huge flood, an uncommon summer flood, and most of the dams along the river had their gates up out of the water, which allows fish to move through the dam. And there's a population of warty-back mussels down in Mississippi and Iowa. We think they swam back up and recolonized. And in Pool 2 now, they're one of the more abundant species in that pool. So it gives us hope for restoration, that if we get the conditions right and the host fish available in the streams, that we can bring mussels back.
Yeah. We have a couple questions coming in. We're wondering if you have a stream stability criteria before you stock new mussels. So they're wondering, for instance, if a river that is over-widened and choked with a bed load likely maybe isn't suitable for mussel species. So how do you identify what's a good spot?
Well, so far we're doing it just through visual interpretation and professional judgment, but there is some interest in trying to develop some hard criteria that are measurable and verifiable. We haven't done that yet, but a bad example for me down here in the southeast where I live would be the river that flows by my house, the Zumbro River. And it's wide, shallow, sand bed load full of dead trees. Banks are collapsing in most places. Where they aren't collapsing, they're disappearing under sandbars that are forming. But to me, that's an example of a place where mussels are not going to do well. And they don't. It's really hard to find a live mussel there. The good example, I think, is below Austin in the Cedar River, where the river bed is pretty stable all the way down. As more tributaries come in, it still hasn't made it unstable, it seems, and that holds all the way down into Iowa, where we go to get our female mussels as hosts. The Cedar River, you would not believe it, but 50 miles down into Iowa, it's a beautiful river, lots of cobble, riffle, pool and stuff, and cobble bottoms, and very stable, and lots of mussels. So, I always think of Iowa as being the big corn desert, and there probably is nothing in the rivers, but that's not really true. I think it's if you move down into Iowa, you get far enough downstream of Austin, the river wasn't impacted by the pollution that came in from the urban areas, and there are no big cities contaminating the river along there. So, pretty cool stuff. And there are some other areas in Minnesota, certainly, but Minnesota River, the lower Minnesota is not in very good shape, but the upper part of the Minnesota is pretty good. It has two really nice tributaries, the Pomme de Terre and the Chippewa rivers that flow into Minnesota River. They have some pretty good habitat still for mussels. They're missing some species we may try to put back there someday too.
Nice. You might not know this off the top of your head, but somebody was wondering if you have data on the species number of mussels in the Schoolcraft River or just the headwaters area of the Mississippi.
We do have some stuff from the headwaters, yeah. And historically, there were about 10 species present above St. Anthony Falls. And we still think one of those may have been an introduced species by moving bait around. But there are areas that are doing really well in the upper Miss watershed. It's largely forested. The rivers are more stable. The Schoolcraft River, I can't tell you off the top of my head, but I would have to look in our database to see what we have from there. We may have survey data from it, quite possibly.
I don't know how in touch you are with other states and what they're doing for mussel research and reintroduction, but where would you say Minnesota is compared to what other states are discovering about their mussel populations and what they're doing about it?
Well, I think our program is actually one of the longest standing programs in the U.S., and our propagation efforts are probably catching up with some of those that started that earlier. Virginia, Kentucky, Alabama all have long-standing and very pretty successful restoration programs going on with propagation. So we use information they've collected, and we have meetings. There's a symposium every 2 years called the Freshwater Mollusk Conservation Society, and it's a 3-day event where we all exchange ideas and present our data and successes and failures. We also have regional groups on the Upper Miss. The Corps of Engineers is responsible for having an annual meeting where we all compare notes on monitoring mussel beds in Mississippi and what each state's been doing. The Upper Mississippi River Conservation Committee has a mussel technical section that Zeb Sechrist from our lab is now the chair for the next couple of years of that, and we intend to disseminate what we're learning and they're doing to all the 5 states in the Upper Miss. But yeah, there's a lot of collaboration, and the other states are moving into this. Nebraska's started doing some mussel stuff. Wisconsin's got some survey work going on. They have had some of that over the years, but I think they may have actually a formal program now. There's a lot of interest emerging. Delaware and Maryland, the program out there on the Delaware River just got a $7 million grant from partially funded by a power company to restore mussels in rivers that flow into Chesapeake Bay to clean the water before it gets to the bay, and then the oyster restoration in the bay is going to do some more of the work. But they're being recognized more and more widely as one solution to polluted rivers, that if we can improve the river enough to support the mussels, they can take it to the next level. And I think there's some need to demonstrate the success of that still, but they're building a big mussel hatchery in Delaware to do just that, to repopulate rivers.
Neat.
Yeah, it's pretty cool stuff. Cutting edge.
So it seems like in your talk, Mike, that kept coming back to that, the whole impact of dams on the connectivity and the movement of the fish that the mussels are so reliant upon.
Yeah.
So I'm just wondering, I know there's been a lot of work in other parts of the DNR to remove some of those outdated dams, and I'm wondering if you've been able to connect with those removals and do some before-after studies, maybe get an idea of how long it takes for some of those mussels to naturally recolonize after a dam removal?
Yeah, we actually have collected some pre-project stuff on several dam removals, including the one on the Minnesota River. And it's on our list of things to do if we can get the funding to go back and see what's happened since then. There are a couple of examples. There was a dam removed at Red Lake Falls, I believe, on the Red Lake River. And the mussel, we had a maple leaf mussel that moved upstream above the dam back into habitat it once had been in before the dam was removed. So we know that was almost within a couple of years we started finding them in the river. So I think it can happen pretty quickly, and there's definitely documented fish movements happening, within just immediately almost after the dams are removed. So yeah, it's very hopeful. I hope that we can do more of that. I'm trying to think of another example, but that's the first one that comes to mind where the maple leaf mussel quickly moved into the river many miles upstream, actually.
Is there anything that kind of on the camp's big wish list of things that you would want to do in the future if you had the funding? What are you guys are you doing what you think, kind of like exactly what you had planned, or do you have like a big project that just needs to get funded?
Yeah, our biggest, most important project at some point in the future is to change our location from where we are to next door to a river, a source of water, a river or a lake. Preferably the St. Croix would be ideal, of course. That'll take quite a lot of money. And build a facility that's like a scaled down version of a fish hatchery where we have the means to acquire most fish we need and the water supply to feed and raise mussels to that size that we want to use for releasing. That's kind of our long-term goal. I guess in the immediate future, we're trying to add more species back to the Cedar River and work with Iowa. Actually, we have one of those species is still present in Minnesota and absent in Iowa, actually two of them. And we can start restoring species missing from Iowa that way. That's part of the collaboration. So we hope to get going on that this summer with a couple more species. We have to do some host work on one of them, the monkey face, to try and figure out what species in the Cedar River is supporting that population. We can use it as a host for the larvae. And, the Mississippi River is a huge place. We'd like to expand what we can do there. It just takes more people and more time. So.
We've got another question from a volunteer wondering, are the mussel survey results available to the public? And if so, where can they find them?
Well, there is some information on our website. There's some concern about divulging information about state-listed species, and I guess there's some concern that somebody might go purposefully try to collect them just to have them in a collection or some other reason. But that's a good question. I think most of the information that's available to the public is either on the website or you can contact someone here at our office. If you have a specific question about your river, we can certainly answer those kind of questions for you. We have a field guide to the mussels and a new poster. I forgot about that. The poster might be very useful for some folks. And we can, you can come here for a visit and take a tour of our lab. Anybody's welcome to do that if they want to give us a call or shoot us an email, one of us. And we have posters here and those books that could be handed out. There was a recent article in the Minnesota Volunteer magazine about our lab that has quite a lot of information in it. But the mussel field guide does show quite a bit of information about each species distribution around the state and whether it's likely to be in the river you're interested in. And we could mail those out to someone if they requested.
Oh, great. So so the field guide or poster, or if someone wants to schedule to check out the lab, they should just email you?
Yeah, that'd be great. You can email me here. It's mike.davis@state.mn.us.
I'll enter it in the little chat thing too, so people have it. Mike.Davis.
Yeah.
State.mn.us.
Yeah.
Great.
So on the website, is there an easy URL that we can give people to go to find basic information?
I'm sure there is, but I don't have it sitting in front of me.
Not handy, yeah, okay.
Well, I guess if I turn the slideshow off, maybe I could find it, but I don't want to lose you guys either.
Don't worry, we can dig it up and maybe
Yeah.
send it out to people after.
Okay. And we do put out a newsletter at least a couple times a year. We're hoping to make it quarterly. And you can sign up to be on the list of recipients for that too.
Great.
That should be on that URL.
Okay. Well, we're recording, so I'll send, once we, maybe by tomorrow, we don't always get the recording right away, but once we get the recording, I can email that out to people, and then we can include some links. We can include your
Yeah, sure. I can send you the links if that would be easier for you.
Yeah, that'd be great, and then we'll get that out to everybody too.
Yeah, cool. Okay, that would be helpful. Yeah, yay for Mussels, huh? Let's hope we get. Well, our patchwork funding is one of our biggest problems. Is we don't know if we have money for more than a year at a time, so it makes staff a little restless, to say the least. So we're hoping to identify some more permanent type of funding. Maybe through Clean Water Legacy, or otherwise we have to keep going back for more and more grants, and managing grants is time consuming too.
So people should tell their legislators to fund Mussels?
Yeah. Don't forget to buy those lottery tickets to fund the Environment and Natural Resources Trust Fund, folks. Although what we get is pretty small, it's definitely the only reason we exist is because of them down here.
Good to know.
Yeah.
Well, thank you so much. This was a really great presentation. I think we all really enjoyed it. And
good. I finished up earlier than I thought even. So.
That's good. We had time for conversation, so that's good.
Yeah, that's good. That's always good. Yeah.
All right. Well, great. So for everybody else, we will we'll send out the recording in a couple of days, and then we'll send out some more information too about Mike and the links to his website, so you can follow up with him if you have more questions.
Great. Thank you.
All right. Thank you, everybody. Have a good rest of your day.
Bye-bye.
Bye.